everyone. It's a pleasure to be here. My name is Fredrik Persson. I'm the CEO of Prostatype Genomics. It's an honor and privilege, actually, to work with this company and especially the product we have developed, which is a prognostic biomarker for patients diagnosed with prostate cancer. I will come into more details, of course, during the presentation. But let me start off with a short background only. This company was founded already back in 2007 as a spin-off from Karolinska Institute here in Stockholm. So we have spent 15 years approximately within the R&D field. And we are also listed on the Nasdaq First North list. Actually, it's about 1 year now, 3rd of November 2020. And currently we have a market cap of approximately 170 million Swedish krona. Every now and then I actually get the question, 15 years, Fredrik, why has it taken such a long time for you to actually reach the stage where you are right now? And the explanation is actually quite simple. And that is when you deal with prostate cancer, you need a lot of follow-up time before you really understand and know if a treatment or a prognostic marker or a diagnostic marker actually works. And the reason for that is that prostate cancer in general is a very slow-moving disease. The challenge is, of course, from a doctor point of view and from a healthcare point of view, that there are also more aggressive forms of cancer that needs treatment as soon as possible. But in general, prostate cancer has a very long-term development. So that's why we need such a long time in order to really know what we're talking about. And we know that. We have a clear focus within the company. Just to show you this one. There are two areas where we really put our efforts in and have done for the past couple of years. And that is, of course, the commercial development of the product as such, and also to continue to expand the scientific footprint. The question is if we ever are going to be finished with a scientific footprint.
Really?
Yes. Okay, sorry. Yes.
You're put on the train,
Thanks. I always get so excited when I get into this, so I start to move. Sorry about that. I will try to stand still. So if we look at prostate cancer diagnosis globally, around 1.3 million men are diagnosed every year with prostate cancer. Our addressable market with Prostatype is around 60 to 65% of the diagnosed patients. And that group is the low risk and so-called intermediate risk group. Our product also works in the high-risk group, but really the clinical utility and the clinical use of this product is mainly in the low and intermediate risk group of patients. That gives us an estimated market potential annually of around 5.5 billion Swedish krona, or 500 million euros. Our plans right now is not to target the global market, rather a more selective way going forward. And this flow chart you see on the screen now represents the diagnostic and prognostic way forward for a patient with an elevated PSA test. I'm sure that the majority of you have heard, or even some of you have been in contact with the PSA test itself. Personally, I took the PSA test on Tuesday this week, actually. So what you see from the screen is that there are 3 separate stages if you look at the arrows on top. First of all, you have the diagnostic part. Secondly, you have the staging part, or what we call the prognostic part. And to the far left, sorry, to the far right from you, the audience, we have the treatment phase. You also see where Prostatype fits in. So I would like to just make it very clear that we are not a diagnostic tool. We are a prognostic tool or a prognostic biomarker. And there is a difference between diagnosis and prognosis. Our test only works when cancer cells are present. We are measuring the gene expression from the 3 genes that are most closely connected to aggressiveness in prostate cancer. And if there are no cancer cells, we cannot measure anything. We have tried, but there's only zeros, a lot of zeros. So we, or Prostatype, should be used when cancer is confirmed. This slide also shows you that the diagnostic phase when it comes to prostate cancer is complex. And what furthermore adds to this complexity is that we are all individuals and individual human beings. Which means that this talk around personalized medicine, you know, is getting more and more evident day by day. You read a lot of it in the papers within different therapeutic fields. And Prostatype is really a very good example of personalized or individualized medicine. Or in this case, a prognostic biomarker. The challenge for a urologist and for the patient is that there is hardly never something that is completely white or completely black. It is a huge gray zone. So what we provide actually is more individualized information to the patient and to the doctor of how to move forward once the prostate cancer is confirmed. The diagnostic today, maybe some of you are aware of this, some are not aware of it, but in general there are three important pieces of information that is used in order to move forward with the patient. First of all, the PSA, which is a blood test, a protein marker. This is, without any question, the global leader when it comes to diagnosis, or the first step of the diagnosis in prostate cancer. Unfortunately, it's a very unspecific biomarker. PSA stands for prostate-specific antigen, which means that it doesn't say anything about cancer, actually. It could be that you have an increased size of your prostate. It could be that you take the bike to the doctor before you are performing your annual check with the doctor. It could be that you have sex the evening before you're going to visit the doctor. All of these examples actually has an influence on the PSA test. And everyone is in the healthcare system is aware that PSA actually is a poor marker, but on the other hand, it's a cheap marker and it's widely used anyhow. What we see in Sweden and also in other countries is that the use of MRI also is increasing. So actually I'm missing one piece of information here on this slide. We should put in MRI. That is not the situation in all countries, but in some countries, in order to further gain information about the individual patient. If the MRI and the PSA gives information to the urologist that, Fredrik, you should come and visit me. Then I go to the urologist, and if he thinks it is appropriate, he will take biopsies from my prostate, inserting needles into the prostate and taking out tissue, hopefully from the cancer areas within my prostate, if I have any cancer. Those biopsies are sent to the pathologist, who, on a subjective point of view, looks at the cancer cells in the microscope and gives a verdict on how he grades the cancer according to the so-called Gleason scale. So you have a subjective part here that is very important, and as you know, subjective means subjective, so there is no, what should I say, definite answer, or that the information you actually is provided with is the correct one. The urologist also uses his finger to feel on the prostate, and that is also a subjective method. So he tries to grade the shape and the size of the prostate, and he puts that information into a T1, T2, or T3, usually. So the consequence of the high level of subjectivity we have in the diagnostic phase, and the fact that PSA is such a poor marker, we know that, means that the consequence actually is that too many men are overtreated with the prostate cancer. And what is the problem with the general overtreatment? And that's the same regardless of where I am in the world. I'm in China, US, Russia, wherever I am. Everybody is aware of this. The urologists and the healthcare systems, the payers, they are aware that we are overtreating prostate cancer patients in general. Again, there are patients that should be undergoing surgery or any other kind of radical treatment as soon as possible, but not the majority. And the reasons are that prostate cancer is, in general, a very slow growing disease. And as a matter of fact, there are very few men who actually passes away from prostate cancer. So being a man, that is a positive. So what is the problem with overtreatment? It's obviously the side effects. And what we see in around 50% of the cases is that impotence is evident, and that is not so fun. Also incontinence in around 20% to 25% of the cases. We also know that there are negative psychological effects, of course, for a man and his partner when a man undergoes this kind of surgery. So again, I'm not saying that not anyone should be operated, of course, but in general we have the challenge with overtreatment. So where does Prostatype fits in? Well, again, we are not a diagnostic tool, we are a prognostic tool when the cancer is confirmed, and we are using the same biopsy that already had been taken during the diagnostic phase. So there is no need for another additional invasive procedure for the patient. You simply extract or take the biopsies from the biobank where it's stored, send it to a laboratory who then extracts RNA from these cancer cells, and then it's okay to use the Prostatype. And what is the outcome and the benefit with Prostatype? Well, I'm trying to illustrate that with the figures to the right. By using Prostatype, we can better stratify diagnosed prostate cancer patients into the low risk group, intermediate risk group, and the high risk group. So instead of having the majority being, in many cases, unnecessarily treated, we can help to better stratify the patients. And this is very important because the group you fall into, low, intermediate, or high risk, depends on the way forward with your treatment. So I will not be too detailed, but Prostatype is the only genetic biomarker today that use embryonal stem cells in the prognostic phase. And what we actually do is that we assess the aggressiveness of the prostate cancer itself. And as I tried to explain to you, we provide information of survival and effects of different kinds of treatments available. And the human being, or a human, consists of around 24,000 genes. But there are only 640 stem cell genes. So the R&D focus from Prostatype Genomics initially was to focus on the stem cell genes. And all the hard work having gone into this product has shown that there are 3 genes that are crucial when it comes to determine the aggressiveness of the prostate cancer. And those 3 genes is what we measure with Prostatype. However, this is not only a laboratory kit. The kit itself is only one part of the whole system. The real value and the real complexity of the product lies within the algorithm we have developed and the huge database we possess covering prostate cancer patients, real prostate cancer patients, where we follow them. I think the oldest one we have in the database is diagnosed 2004. So that gives us 18 years of follow-up time now. The information provided by Prostatype, the gene expressions from the 3 genes, together with the PSA, together with the Gleason score, and together with the T-stage, is entered into our web-based Prostatype web system. And you can see to your right, out comes the report for the doctor and patient to look at, where you can see if by using Prostatype you are classified as a low-risk, intermediate, or a high-risk patient. So again, this is not only about the kit, laboratory kit. There is so much else surrounding this product. The question is, does it work? Of course. That is always the case. I've spent 30 years now in this industry, and in the end, you obviously need to show clinical performance for a product. And you have to fulfill a clinical need. And the clinical need is definitely the smallest problem we have. Everyone says that, yes, more or less daily, I'm struggling with understanding really what to do with this particular diagnosed prostate cancer patient. And the good thing, and what is so inspiring, is that this is a global phenomenon. It doesn't matter where in the world I am. So if you look at these bars here, the black bars, they represent the current golden standard. And what we have done is that we retrospectively have looked at those patients. It's actually from the Stockholm area again. And we have used those patients' biopsies and made Prostatype procedure with those biopsies. And where you can see the added value by using Prostatype when it comes to the classification of the patient. So that's fantastic in Sweden, that we have the access to biobanks and tissues being able to perform this kind of procedures and R&D. And not surprisingly, you can see that the highest added value is in the low and intermediate risk group. This is internal data, a compilation of all the data and all the information we have on our files. But the real test is when you use the test in reality.
What does 0.72 mean on that slide?
0.72 means the accuracy of the decision taken initially on the patient. Did the patient actually die, or did he not die? So it's accuracy numbers, you could say. So the real test is, of course, in reality. And we have performed a large study in Skåne, performed by Associate Professor Göran Ahlgren, where I will not dig into all the numbers here, but you can see that actually the results from this study was better than we have been able to prove ourselves. 35% of the patients were reclassified by using Prostatype. In 2 out of 3 cases, yes, we confirmed the treatment decision that was taken. I would, however, like to draw your attention to one part here, and that is the D'Amico, the column to the left, where you say intermediate, and then the next column says P-score. There you can see in the following column that 37% of the patients initially diagnosed as intermediate were downgraded to low risk. And that is super important for the individual patient. Because low risk, then there is not a radical treatment in general. It's what's called active monitoring. meaning that the patient comes back to the doctor every year or every half year, just to check the PSA level. So we can also see that also in the high group, there were patients that or by using Prostatype, were downgraded to low risk. And you can ask yourself, how is that possible? That's at least what I did. I was quite, stunned when I saw these numbers the first time. And Göran Ahlgren, of course, looked into these, I think it's 13 patients, yes. All of them were classified as high risk, simply because of one thing, the urologist's finger. Because if you're classified as a T3 patient, automatically you run into the high risk group. This shows the complexity and the problem with subjectivity when you're diagnosing and prognosing a prostate cancer patient. So we are happy that we can contribute in this way. Looking at, I'm leaving the product now, moving into kind of a summary when it comes to the stakeholders around Prostatype. First of all, of course, we do have the payers, the landsting or county councils or private insurance companies. And by using Prostatype, of course, by reducing the number of radical treatments, of course, there's a cost saving in that. From a laboratory point of view, the laboratories are not our customers. They are a doer. They are just performing the test. For them, it's easy to implement Prostatype, as from a technical point of view, it's not rocket science. It's a PCR test. And the majority of labs are able to perform a PCR test. That means that the entry barrier is very low towards the laboratory. For the pathologist, it's also interesting, as our objective way of measuring and classifying the patient balances the subjectivity of the Gleason score performed by the pathologist. And for the urologist, of course, the urologist owns the patient. And the urologists, they are our customers. They are our focus. So for the urologist, we are a treatment decision support. We defer repeated biopsies. And as I've tried to explain to you, it reduces the risk for over or under treatment. The most important player in this picture here is, however, the patient. And I'm talking to so many prostate cancer patients, and I really, really suffer with them. There is a high grade of uncertainty when you have received the diagnosis of prostate cancer. What should I do? Will I die? Or will how serious is this? So there are many discussions, many phone calls that really is, from an emotional point of view, fantastic, I would like to say. So the patient is also important for us, and that's why we are here, really, in the end. Our plans for actually succeeding with Prostatype follows a plan that is not unique in any way. This is what life science companies in general needs to do. First of all, to prove that the clinical function of the test. We have done that. We are in processes of further retrospective studies, but we already have results from retrospective studies. Another interesting information will be published in Cambridge, in the UK, in December, performed at the Akademiska Hospital here in Uppsala. Our focus right now is on point 4, which is the pre-reimbursement sales activities. And that is guided towards the private market. And the private market consists of private insurance companies, private out-of-pocket paying patients, and other private players like hospital chains and hospitals. Because reimbursement, we are working with reimbursement, but we cannot wait for reimbursement. We don't want to wait for reimbursement, partly from a patient's point of view, but also from our point of view, of course. So reimbursement will take time, but we are working with it already. So The business model, if you start from the right when you see this picture, as I said, the focus now is on the private market, and the price, average sales price, is around 22,000 Swedish krona. That could sound expensive to many, but if you compare with the cost of overtreatment, for example, it's very cheap. Our key player in this screen here is the distributor. We are not planning to build a huge own sales organization. We are going to work together with the best possible distributors in the selected markets. So the distributor will, in the end, invoice 22,000 krona, and to us, he will pay 6,500 Swedish krona. That gives a huge margin for the distributor and the motivation to really work with our product. That's not the whole gross margin, because there is a cost for the lab test, around 2,500 Swedish krona, that someone has to pay, and that will be the distributor. And in certain countries, you also have to pay for the doctor. So this is how the business model looks like. The price of 6,500 Swedish krona gives us, Prostatype Genomics, a gross margin of around 90%. We are happy with that. And just briefly before I stop, some interesting developments are happening on a daily basis in this company. And recently, what we have done is to select it after a long and hard work, distribution partner in Spain, whom we believe very much in. We have also initiated a distribution and lab cooperation in the UK. And we have started and initiated a commercial and R&D cooperation in Switzerland with a company called Proteomedics. And yesterday, and I think today, we have sent out press releases as a consequence of this cooperation that we have started to work in Italy and Germany. So this Proteomedics, just briefly, it's a privately owned Swiss company. They have a ready-to-go product. It's sold already today with a very strong scientific footprint. From our point of view as Prostatype Genomics, we will benefit from their contacts and their work when it comes to the commercialization of Prostatype, and we will, as a combination, be more attractive towards the top-end distributors whom we are targeting. And the products complement each other in a very, very nice way. I don't have the time now, but it's also super interesting from an R&D point of view, as this collaboration will enable us to enter into the world of multi-omics. And multi-omics is a combination of two different omics. So various blood markers or biomarkers from different sources, for example, blood and tissue. So just to be clear here, Proclarix is in the diagnostic part, pre or post MRI, and we are in the prognostic part. And I'm not going to bore you with how good we are and with everything we have achieved. But as you know, I mean, there has been a pandemic, but we have actually managed more or less to fulfill all the milestones we have set up when we performed the IPO. So I'm very happy to see that continuing. Thank you very much. I hope this was a good presentation and that I've been able to focus on our two main areas: commercialization and strengthened scientific footprint. Thank you.
Thank you, Philip. Excellent. Okay, some questions.
Yes, please.
It seems the benefit is 75% reducing overtreatment, and 20%. It must also save someone not being diagnosed that needs to be treated. But your focus is stop overtreatment. Is that correct?
Well, I'm not quite sure I understand the question completely, Lars. But
the major benefit is that you stop overtreatment, yes, and reduce the side effect, negative side effect.
Perfectly correct. And you also have, which I think is the 25% you're referring to, a diagnosed cancer patient that with the normal tools is regarded as a low-risk patient. If our test shows high aggressiveness, yes, that is a very strong signal that, oops, this patient should also be treated. But the main problem is overtreatment. You're perfectly right with that.
Yeah. And that is because it makes perfect sense. The treating team, doctor, would say better safe than sorry. He wants to err on the side of overtreatment.
Perfectly right. Perfectly right.
Okay.
And that's why we have the overtreatment today, as you say. I mean, okay, there is a lot of uncertainty, but I mean, to be on the safe side, let's perform a surgery. And I can understand that. If I was a urologist, I would probably reason in the same way. The frustration, I think, is that, okay, the current tools we have, they are too much based on subjectivity, and they are simply not specific enough. What we are talking about here is not a huge national care program saying that you should be treated in this way and I should be treated in exactly the same way. We are looking at the individual patient and the individual patient's possibilities to undergo surgery or not undergo surgery.
Okay. So,
personalized medicine.
Yeah. So to simplify a bit, your reason to be, Prostatype Genomics,
yeah.
reason to be is less impotence, less incontinence.
You could draw it, yes, you could say that. Better quality of life or improved quality of life for prostate cancer patients. That's how I would phrase it.
Yeah. Sounds good to me.
Yeah. Okay.
And this is, you have looked at, through some means, you have looked at 24,300 genes,
no.
and you decided on these three.
No, we pretty quickly understood that that's not possible. But it is possible to focus on a smaller number of genes, and actually the stem cell genes, they are 641, as I said. And of course, behind that thinking is the basic thinking, the original thinking, that stem cells as prognostic markers or biomarkers should be something interesting.
I'll hand over the mic here.
Yeah.
Well, this is really important to make something for this overtreatment, but I would ask about the Stockholm scene. You now in Stockholm have a spread of the Stockholm 3 test with the same aim. Can you comment on that?
Well, first of all, I'm not quite sure we have the same aim. If I may, from my understanding, I'm not an expert on Stockholm 3. I've heard about them for sure, but from my understanding, they are more here. I've read some interview the other day where the founder said that they are here to replace PSA. And that's a completely different segment of the market compared to where we are, where we're talking about diagnosed cancer patients. So we are much later in the process. Is that okay with you?
I can say if they are right, they do the selection earlier, and then your figures in the last must be quite different. I mean, how many you will save?
Again, I'm not an expert on Stockholm 3, so I'm sorry, I cannot really comment on that.
Can you explain the regulatory process? Do you need to be approved? Have you been approved by the relevant authorities, or do you need approval at some stage? And how are your results? Are they good enough, or how far are they from being approved by the authorities?
Thank you very much. It's a very relevant question, of course. Our product is CE marked, meaning that the product is possible to buy and sell, especially from our point of view, within the whole European Union. So that's already in place. When it comes to the US market, which also is interesting, of course, we are not planning to go via FDA approval process. It takes too long time and it's too costly. So we will look for what is called an LDT path forward, a laboratory developed test. So that is a much quicker route, and actually the competition we have, US-based companies active in the US, they are also LDT registered products, so they are not approved by the FDA. So yes, to answer your question, Europe is fine, When it comes to China and the US, markets where we also are active, we need some kind of approvals in those countries in order to be able to commercialize the product. Correct.
One last question, short answer.
Yeah. We discussed in the intermediate, if I were to have a cancer that shows none of these 3 gene expressions, how stable is that? Can I expect the cancer to stay in that state for a number of years?
What you usually talk about is 10 years survival. We are also talking about 10 years survival from diagnosed prostate cancer, meaning that you or me as a patient will not die from prostate cancer. What is a prerequisite is that we actually have cancer cells evident. If we have benign cells, not cancer cells, and we test prostatype, there are zeros. The test do not work in normal healthy cells, so to speak.
But it's not that these genes pop up a week later after I've taken after you have made your your research.
No. When I fall back to what I can say, and that is we're talking 10 years survival. Yeah.
Thank you.
Thank you.
Prostatype Genomics.
Thank you.